How can we isolate genomic DNA from whole blood by magnetic bead based method? The steps include blood lysate preparation, DNA binding, DNA washing, and DNA elution. Blood lysate preparation. The lysate preparation was initiated with RBC lysis steps by adding the whole blood to a sterile 1.5 ml MCT tube containing blood lysis buffer 1 and vortex for 2 minutes. After a fine vortex, add solution A and invert mix the tube 4 to 5 times and incubate the samples for 5 minutes at room temperature. Centrifuge at 8000 rpm for 2 minutes at room temperature. Discard the supernatant and repeat the step once again. Finally, WBC cells were pellet down. The WBC cells were lysed by adding blood lysis buffer 2 to the pellet and resuspend by pipette mixing. To digest the protein and remove contamination in lysate preparation, proteinase K is added and resuspended by pipetting. Incubate the samples at 56 degrees Celsius for 10 minutes. After incubation, transfer the lysate to a fresh 1.5 ml MCT tube. Next step is DNA binding. The nucleic acid was isolated from the lysate by adding blood MagGenome and gently invert the tube 10 to 12 times to mix properly. Do not vortex. Incubate the samples at room temperature for 5 minutes. Place the tubes on a Magna stand until the solution becomes clear. Carefully discard the supernatant without removing the tube from the Magna stand. Ensure the magnetic nanoparticles are not disturbed. DNA washing. The nucleic acid bound to the surface of the magnetic nanoparticles is carefully washed to remove impurities by adding blood wash buffer 1. The tubes were removed from the Magna stand and resuspended by thorough pipette mixing to ensure complete dispersion of the particles. Place the tube back on the Magna stand for 30 to 60 seconds till the solution becomes clear. Discard the supernatant without removing the tube from the Magna stand. Ensure the magnetic nanoparticles are not disturbed. The bound nucleic acid was surface washed by adding blood wash buffer 2 and gently invert mix the tubes 5 to 6 times without removing from the Magna stand. Discard the supernatant without removing the tube from the Magna stand. Surface washing steps were repeated once again. The tubes with the magnetic nanoparticles were air dried without removing the tube from the Magna stand for 10 to 15 minutes without over drying them. Next step is DNA elution. The nucleic acid bound to magnetic nanoparticles were eluted by adding blood elution buffer. Remove the tubes from the Magna stand and resuspend the magnetic nanoparticles by pipette mixing thoroughly. For efficient elution, incubate at 56 degrees Celsius for 5 minutes without intermittent tapping. Place the tube back on the Magna stand until the solution becomes clear. Carefully transfer the supernatant containing DNA to a sterile 1.5 ml tube without removing the tube from the Magna stand. Ensure the magnetic nanoparticles are not disturbed. Store the isolated DNA at -20 degrees Celsius. Quantitative and qualitative assessments. The results of numerous molecular screening and assay methods often rely on the overall quality of the genomic DNA input. Downstream application of DNA is purely dependent on how better we are able to qualitatively as well as quantitatively isolate the DNA with a robust and affirmative analysis. Nowadays, UV analysis of DNA has been benchmark method for rapid quantification and assaying purity of sample for various purposes. The heterocyclic bases of DNA are aromatic and absorb in the ultraviolet region of the electromagnetic spectrum. It is noteworthy that lambda max for all Watson-Crick bases is between 250 nanometer and 280 nanometer. So, following Lambert-Beard's law, it is possible to estimate the concentration and quantity of the oligonucleotide in solution. The isolated DNA were quantified using spectrophotometric method. Double-stranded DNA application was selected. Blank was run using 1xT buffer. The DNA samples were measured by loading 2 microliter of isolated DNA sample and click measure to quantify. The graph interprets the quality and quantity of the DNA. Nucleic acid have maximum absorbance at 260 nanometer wavelength. Purine's absorbance maximum is slightly below 260. Pyrimidines maximum is slightly above 260. Purines have a higher molar absorptivity than pyrimidines. Therefore, the absorbance maximum and absorptivity of a segment of DNA depends on its base composition. A pure DNA sample, the A230 is to 260 is to 280 should be around 1 is to 1.8 is to 1. As a guideline, the A260-A230 ratio should be greater than 1.5, ideally close to 1.8. Concentration of double-stranded DNA is equal to 50 microgram per ml into dilution factor into absorbance at 260 nanometer minus absorbance at 320 nanometer. Agarose gel electrophoresis. Gel electrophoresis is a technique used to separate DNA fragments according to their size. DNA samples are loaded into the well at one end of a gel. The agarose gel matrix is porous and acts as a sieve through which the nucleic acid molecules migrate and an electric current is applied to pull them through a gel. DNA fragments are negatively charged, so they move towards the positive electrode.